Hello, and thank you for watching this presentation by the American Iris Society. Please support the organization by becoming a member. Go to irises.org and click on Join. Thank you.
I'm covering broken color and I say and variations because there's a lot of different things going on in here, and it's pretty hard to really describe what's going on with broken color irises unless you cover some genetics and a whole bunch of other stuff. And there's a lot of hybridizers who are interested in this program, so I'm going to put a number of information in about genetics, and I'm trying to explain it as best I can. But if you're not interested in genetics, don't worry about that part too much. But I know for the breeders and the hybridizers are very interested in that sort of information, so it's in there for them primarily. I'm located in Ospringe, which is just outside of Guelph, Ontario, but I'm actually located south of Salem and Oregon with all of the things. And normally my peak bloom would be about June 8th, but we're in Salem is in peak bloom May 19th. So with my so this is what my seeding patch looks like. This is one from a couple years ago, and I'd like it to look like this every year, but it doesn't. But this shows you one example of when I had a really nice bloom in the seedling patch. This, I think, is probably the third year of this seedling patch, and it kept, I take my selections out of there, and everything else goes onto the compost pile. This is a greenhouse that I have, which I do some breeding in, and it's total of 120, I guess 130 feet long, and I've got what 80 feet enclosed there. This front part right here is a heated part of the greenhouse, and this shows you the irises in the bottom corner there, shows you what it looks like when they're in bloom in there when I go do the breeding. So inside I'm about equivalent to Oregon, so I get 2 periods of bloom there to breed with. But I find that my climate is so harsh that I have difficulty keeping some of these nice irises up from Oregon and California and other places like that healthy enough to bloom and be able to use for breeding. There's no point bringing them up if I can't use them for breeding. So that's where the greenhouse is. I plant in there my own irises to use with the ones I bring in, so I get the chance that. Here's a picture of the house from the road, and you can see the greenhouse grows in there, and nice crabapple tree in bloom. I also have a number of 30 beehives on there. They're not my beehives, but I do get a 5 gallon pail of honey off them every year because from the owner of the beehive. So it comes in and takes care of them and gives us a nice pail of honey at the end of the year. This gives you a summary of what I'm going to talk about in today's presentation, some of the history, some of the effects of cold development, frost development, ABC of flowers. So what is actually composed of a flower and how this can go awry and create some very mixed colored flowers. Effects of roundup, effects of viruses, a little bit of talk on genetics here in terms of recessive genes, and charisma, chimera, which is a sort of what happens when you get these patches on it. Now, broken color irises or colors of mixed color have been around for a very long time. And this shows some of the very, very early ones from 1840, Victorine, Floralee, which is widely distributed, and you can find that in quite a few places from 1909, and Japanese, which is actually a double, but it presents with these mixed colors on the petals. Kaleidoscope, which has been around since 1930. W.D.R. Dykes, and I'm going to speak specifically about that one later, and Joseph's coat. Corsage, which is an interesting one which is shown up, and unfortunately that sort of mixed color with the yellow on white hasn't been captured in breeding program. Humoresque, which is one that Keith Keppel introduced in 1962, and he didn't do any breeding with that specific for that color because he didn't quite realize what was going on there. And by the time he realized it was sort of past its prime time. Minnesota mixed up kid, which is a kind of a, no one knows quite sure where it comes from, but it's been around and has a very interesting color in it. In terms of the history, Alan Ensminger was the first person who really did it, focused on doing breeding with it. And he had a couple show up in his garden. He's quite intrigued with that. He asked people to send him any broken color irises and cultivars, but he actually didn't receive any. But he undertook a breeding program, and he was quite good because he would offer this package of broken cultivars, which are seedlings, which he sent out to people to use for breeding because he was strongly encouraging that. And I got that package, and which included one of his irises, which he later named. These are some of his very earliest ones: Dominicus in 1974, and Doodle Strudel, Batik, which is very well known of his, and I think that's located in a huge number of gardens. Autumn Years, which I got from Alan as a seedling, and he didn't actually introduce it. Someone else introduced it for him, or put pressure on. I can't remember which is correct. And that's a very, very intriguing iris. Interesting colors. Brindle Beauty and Mera Tremena are two more of his irises, which are very widely distributed and very well respected. Batik, of course, is everywhere, and I just suppose I got that double there. It wasn't supposed to be. And isn't this something? Painted Plick and Peach Jam are two more of his, which are very well known and used a lot in hybridizing. Purple Streaker and Very Varied, two other very interesting ones, which are not quite as well known and distributed as some of the others on there. Now, Brad Kasparek is another big name in doing hybridizing with it. And that's Brad in his zebra vest suit, which he wore to all the conventions. And that's a copy of his catalog there. I took a scan of a page of his catalog. And quite outgoing and quite a gregarious fellow. William Falcon is one of his, and that's well known and well liked. And the Wildebeest, again, another one which is in a lot of gardens around. Baboon Bottom. Brad liked naming things with weird names, and Baboon Bottom certainly got a lot of attention. And I know a lot of people were talking about the name, and some loved it, and some bought it just because of the name. And correspondingly, a lot of people refused to grow it just because of the name. So the way it goes, names can sometimes be a very important part of naming your iris too. Tiger Honey, a very nice, very attractive iris. Anaconda Love. Gnu's Flash is another one of his, which is very well known and very well liked, but unfortunately it's not very cold hardy. And I've had it survive for me like maybe 3 years, and then it disappears. And I get it again, and it lasts for about another 3 years and disappears. It's just one of those things. Draft and Kinkajou Shrew, two of his which are not quite as well known. Flamingo Grinko and Spice Taggers, which are very interesting. One nice thing about the Spice Tagger is that you do have that nice yellow background with the red on top of it, which I think is very, very striking. And there's not a lot of broken color irises that show that sort of pattern. Now, I'm putting Paul Black in here as a significant hybridizer of broken colors. And one of the main contributions that Paul has done is getting away from the placata-based broken color to the non-placata. Because all of the ones that I showed you with Ensminger and Kasparek are basically on a placata background. But Paul has developed a whole line of his which are on a background which is not placata. And I'll be speaking about the differences of that and more specifically in part 2. So Big Break and Seek the Unique, two very nice, attractive irises. Die Laughing, which is one I've used quite a bit in my hybridizing because it's a very nice, strong grower. Wild Streak, which has that sort of like, it's very nice as pics of apricot in the background with purple streaking on it, which is, I find that mixing those colors up is very attractive in these. Zip Zing Zowie and Broken Pattern. And two of the more newer ones from Paul Black, which give very, very striking colors. And the nice thing about Paul Black is that he's getting in nice big flowers, nice stalks, good bud count. I'm putting up George and Mike Sutton basically because of this particular iris, Peggy Ann, which has proved to be very significant in terms of breeding. And one of the significance of that is that how much there's heavy streaking. So you have basically, it looks like it's white streaking on purple, but actually it's purple streaking on white. And I'll explain more of that during the genetics, particularly in the second part, when I go more into the genetics of what's actually happening under the hood with these. And Silver Streak, which is a nice horned space age iris with those mix of colors. Purple Marble and Break the Barrier, both of which have Peggy Ann in their background, giving you that very, very dark, heavy streaking. And one thing you know on these, I want to point out on that is these are placata base, but very nice, wide, well-formed flowers, good substance, good texture, good color, good markings on them. Broken Record and Break It Up. Now this Spiced It Up is one which I think is a nice breakthrough because you've got that yellow, nice bright yellow background with the heavy streaking on it. And this is a seedling which is from Spiced Up and showing you that you can carry that genetics through into other irises. David Toth and his daughter Iva have really picked up on the breeding, and they've introduced an incredible number of broken color irises with a whole mix of color and pattern on it. Blueberry Smoothie. And this one that David is very particularly proud of is Sheer Panic, and he's quite pleased with that one. And it won a AM award last year or a year before, I'm not quite sure which, but certainly on the award chart, and one that I would recommend that people try. I haven't tried it myself, but it seems to be an extremely good iris. Early Morning Rays and Exceeding Expectations, two more of his. Jelly Splatter, which I quite like because you've got that purple splash there on the white. And I suspect this one is probably a non-placata background, but it's hard to tell sometimes when looking at these irises in the background because there's certainly the placata irises get mixed up an awful lot in a lot of other irises. So it's hard to tell when they're there and when they're not there. Krakatu Katie, and a very interesting one because it has a blue background. So it's a dark red, or dark purple on a blue background, Shadow Illusions. And so I think that's Iva Toth, but I mean, unfortunately, I can't see that part of my slide right there. Jelly Splatters, Abstract Dream. Oh, I seem to have been backwards there. Now I'm going forwards again. Sorry. Havana Cabana and Making Lemonade. Sleep Deprived and Butternut Try It. Ava seems to focus more on the IB, the intermediate bearded irises. So there's a lot that she's introducing in that, and David seems to be doing more of the tall bearded iris. Another factor that causes mixed colors in iris is frost damage, which can affect the texture and form of an iris, but can also affect the color and can produce colors which are sometimes mixed up and sometimes look like broken colors. Here's two more examples of that where you get the frost damage. This one doesn't show so much in the color variations, but there's some color patterns and changes on that one. This one shows a very good example of what happened. The same iris power down without frost and with frost. You can see the sort of the resemblance to the broken color iris just there with the frost damage. Another thing that we get sometimes is a variation in terms of weather. So leopard print, and this is what it can look like or looks like in an awful lot of gardens. And this looks like when it's a sort of gets its maximum spotting, but it seems to be related to having cold nights during the development. And this is one thing that happens with a number of these is weather-related effects. And you can get more of the spotting with cold weather and less with warm weather. And so another thing that comes up with iris is developmental errors, where the plant is actually making a mistake. Because when you have the petals and falls of iris, and these are often in different colors. And if there's a confusion in the plant in terms of what is a fall and what is a standard, then you can get these mix of colors. So sometimes petals can be identified as falls and falls can be identified as petals, and it causes mixes of that. Now I'm showing you a little bit about the genetics, and it's called the ABC of flowering, because it looks at it in terms of the two genes. And so you have the B and the C together, you get the stamens and the you know, there. But when you get the B and A, you get the petals. And when you have just A on the sides, you have the sepals, which are basically the falls. And then the A and B gives you the standards. But when you get that mixed up, so here you have a flower that, if you notice, it has two falls and two standards. And one of the falls is confused as whether it's a fall or a standard. So the standard color is now shown on the falls, and that's a developmental error. Now, Laura Lee is one that shows that sort of mixing of color just with its standard shape. So what's happening in there is that there's a confusion here between just being an A and an AB. So you get these sort of mix of colors on it and the mixed up on it. So the plant is not quite sure whether it's a petal or a fall. So you get the color of both the petals and the falls, and there's kind of like random mixed up, kind of like with a calico cat in many respects. Here's two irises from, my seedlings. They're just showing abnormal color in the garden. So I just sort of grabbed the pictures of that just to show you. And again, this confusion, this one has one standard, three falls, and but all three falls are kind of thinking that they're half standard and half falls. And then you get this same sort of mix up. Another example of that, and this is a seedling that one year showed itself as a flatty, and it showed this mix of color. Next year, it showed itself as a normal flower. So I was hoping it had a nice little flatty there, but it kind of disappeared. So we also get this with flat irises. So this is where you get the standards are falling down. You end up with six falls, but out of the falls that were previously standards, end up with these mix-up of colors. So you get this mix-up of random colors. Clydesdale, Fiasco, two more of the doubles or the flatties, where you get that mix-up and you get that sort of random distribution of color. Another big issue that we have, and this comes up time and time again, and this is a very serious issue, and we have to sort of pay close attention to it because it's can be a real problem for us, which is mosaic iris virus. Now, I'm showing you here the severe iris mosaic virus, and there's actually 6 different mosaic iris virus have been identified, and these are show various effects on the plant, and this is showing you some of the leaves on that. Here's showing you again a more detailed picture of the leaves, and what you got is that sort of streaking on the leaves. We have the dark streak and the light streak, and this is severe iris mosaic virus, and it can be very hard to identify. There is a genetic test for it, but it's not something you can get at most plant pathology labs. So but they can send off for it, and they can send a sample in and get a genetic test on it. The thing with the virus is that it's an infection, and it spreads out from a point of origin, so it spreads out in all directions randomly. This is important because it helps us in being able to identify the difference between a broken color pattern that's called by a virus versus one that's called genetics. And I'm going to show some examples of that later in a couple of slides. The tulip mania in Holland was a prime example of a mosaic virus. it's probably not quite the same mosaic iris, but what it does is it shows that broken color. But one thing that the virus does, besides breaking the color of the flowers, is it affects the strength of the plant. So what's made these so valuable is that you get very few of the bulbs are actually increasing. And so this shows you sort of a graph of the increase in value, and it sort of went up to a great big peak, and it kind of like crashed all of a sudden. So there's a financial disaster on there too, because people invested a lot of money in these. I heard one interesting story that I relate to is that one of the people had a very expensive tulip bulb, and he had it on his ship, and he's going to take it, and he turned around and it disappeared. And it turned out that one of the sailors had thought it was an onion and picked up and ate it. So he wasn't very happy with that. Now, here's as app. Now, this picture over here on the right is a picture that you see very often with as app. And people have started to think of as app as a broken color. But the picture over here on the left is what it looked like when it introduced. And that is, I got the iris directly from Alan Insminger. And I was following it as it's sort of presenting, and it goes through the awards and attention. And that's how it went. And it's a very attractive iris, nice late pale blue, nice shape, nice, beautiful color. But this is what you see when it's a virus. And you see these spots. They're kind of like random. But notice also on here, you have texture, you got sort of wrinkling. The edges are not smooth, so you can compare these irises side by side. Nice, clean petal margin, broken petal margin. Solid, nice color. The spots are sort of like there's splashes. They're not kind of like it's kind of like you dropping a spot of coffee or something onto a blotting paper. W.R. Dykes, and I'm not sure there's anybody who's got the original Dykes. And my, there's a blog in terms of World of Iris on there, and it's published in March 31st of 2012. So you can look that up. And I just try to remember the name of the person who wrote that, but thought I had the name on there, but I lost it now. And he mentioned this too, and that the original W.R. Dykes, when it came out, was described as an astonishing, bright yellow, well-formed, well-marked, and everyone thought it was rushed to use it in the breeding. But then what happened is that it showed up in this sort of broken color form. But unfortunately, this broken color form is actually the mosaic iris virus. You can see that in terms you look at the petals, and you can see that the texture is a real problem. There's all sort of broken. The spots are not in any sort of a typical pattern. They're kind of like, again, water spots that have leaked. So unfortunately, we probably don't have the original W.R. Dykes around. We just have the one with the virus, which is unfortunate. Now I'm showing you these two examples here, because one of the things that happens is that sometimes it's hard to tell whether we actually have a broken color that's genetic or an iris that's actually a virus. Now, it's been suggested to me by a couple of people, this one is probably a virus. And there are a lot of signs that there's a virus. These spots, especially on the standards, are kind of random. They kind of look like watermarks rather than triangles. And again, the spots on here are not as clear as it could be, and you have texture problems. This is a seedling of mine that showed up, and it very clearly is a virus one, because again, the spots are kind of like they're washed. There's no clear margins on the spots. It's kind of like blending spots. So again, that's something we have to be watchful of, and sometimes it's not exactly clear. With a virus, we get an uneven texture of petals. You have weak texture. The spots have no clear margins. They're not in a triangle shape, whereas in the genetic controlled ones, they're usually in a triangular shape or triangular pattern. Petals have uneven and irregular margins on the virus ones. There's no reversions in that sense that you don't have a case where the flower has actually reverted to a non-broken color form, and I'll be showing that a number of examples later on that, but basically in part 2. The virus shows itself more in warm weather and less in cold weather. Genetic controlled ones has the colors, the splashes are in wedges, passed on genetically. You have even petal margins, you have even texture of the petals, and weather has no particular effect on expression. When you're judging, for the most part, I put this in there because a lot of people asked about in judging. So when you're judging the broken color irises, you're using the same sort of judging standards as you have in the manual, the same judging standards for the show bench and the same judging standards for the garden. But one thing I want you to watch for, and this is critical, because not supposed to be a broken color. If it's diseased, it should be disqualified. The broken color, focus on the texture and symmetry of the petal and the petal structure. So are the spots nice and clean? Do they have the clean margins? Are they in wedges? Or do they look more like you kind of dropped some dye on top of a blotter and it kind of like spreads out in a second? Is this spot directed or is it kind of random? So again, if you're judging as apt, I think basically, in terms of my opinion, the one here on the right, the ones you see of as apt, I think I disqualify that on that as being very clearly diseased. And here I'm showing you an example of squid squirt. And you look at these two pictures. The one on the right here, nice even petals, nice even margin. The spots are sort of nicely defined edges. But you look over on this one, you see a lot of spots that are not clearly defined. They're kind of like the dye on a blotter type of thing, where they're spreading out in all directions. Whereas you see what some here that are spreading in an even direction, but so you have to watch that carefully on that when you're judging. So that's something that you always have to watch for and be careful of. And in terms of judging, does it show nice clean petals? Does it show a lot, nice texture and so on? Now I'm showing a section with roundup because every year I get there's a number of people during bloom season show pictures of irises that have been affected by roundup, and they're quite sure they have a broken color iris and they're quite fallen in love with it. And when you try and tell them that it's roundup damage rather than being actually a mutant or anything else, they get really upset until they actually start. I had a number of people who insist that it's not, and I say, do some research, check your neighbor, check everyone else in the family. And when they do that, they'll come back, oh, my husband used Roundup and didn't tell me about it, got close to it, or the neighbor sprayed some Roundup. But what happens there is that Roundup is this very unique chemical. It's a group 9 herbicide, because all the herbicides are classified in terms of groupings and that effect, in terms of how they work. And what Roundup does is it affects the EPSPS enzyme, or what that does is it affects the production of amino acids. And the amino acids are used as the building block of anthocyanin. So one of the first things that we see is a disappearing of color, because you get less of the anthocyanin disappearing, and with more of the dosage, you get affects the form of the form and the size of flower as well as the shape. But it's usually a temporary effect, because when it's hit the flower bud, and it doesn't usually at that time, it hasn't affected the increases, so that it's sort of like a dosage effect, a headache. And often a number of the flowers that come out later on the flower stalk will be normal in terms of flower shape. Here's trisocine, which is amino acid. Now all of the aromatic amino acids have this benzyne ring in it here, which is actually crucial because definition has 3 benzyne rings in it, and it's and without those, and it's using this tyrosine to build these benzene rings. So without that benzene ring, what happens? You get less and less of the definition. So here's showing 2 irises affected by Roundup. These are sort of light dosages. You can start seeing the petals start to curl on that. This is one that you often see show up or in the spring, and people are all excited. But look at this very gorgeous iris, and you get all sorts of oohs and ahs. And this is one where you have to tell people, sorry, that's not a new iris. That's not the way it is. And well, that's life. Here's another one. Two more showing one with a mild effect and one with a severe effect. And this is sort of the curling that you get with a high dosage. And very high dosage, you get this curling. But even with this one, you look at this, the flowers as they come out later on the iris stalk can actually be fairly normal looking. And if you're lucky, if it has lots of flowers, some can even be absolutely normal. I'm going to talk a little bit about the flower pigments and iris because without understanding the flower pigments and iris, it's hard to understand what's going on and what you're looking at. And we have two sets of pigments, the carotenoids, which are the yellows and the pinks, and the anthocyanin, which is your basically your definitin, which is blue. And each of these are located in different parts of the plant cell. The carotenoids are in the cell wall and chromoplasts, which are modified plasts like, you know, chloroplasts, like, like your green in the iris in the leaves. Anthocyanin is in the cell vacuole and it's water soluble. And each pigment is controlled under different and independent genetic controls. This is a slide of an epidermal peel of an iris flower. My own slide here. And when you peel that, you can look at it. And these spots in here are actually your chloroplasts with oil dropules in them, which is what gives it a color. And there's many spelled, and these are all in. I'm going to show you a picture of a cell later on and showing. This is the pathway for all of the carotenoid pigments. And the carotenoid pigments follow us. You could take one gene, and the parts there in the capital letters you see on the side are the gene. This letter in here is the actual chemical. So this gene changes that chemical to that chemical. This gene changes from there to there. This changes from here to here. This changes from here to here. So this is your pink, and then you have various yellows. So I'm going to come back to that later on. Here's a slide showing all of the different. These are all pigments that are showing up on the slide previously. So there's the one with no carotenoid pigments. This is your orange pigment, pink, and you have various shades of yellow, which are actually all can be there in the iris. You have one or another of these carotenoid pigments. Delphinidin is the blue pigment in iris. It's a type of anthocyanin, which is a bluish purple, not really a true blue. So when you get a blue looking color in the iris, there's some modifications going on. And it could be part of this. Part of it is the structure of the flower and some modifiers. Definitive is particularly darker in cold weather, particularly when if you have a bud that's developing and it's shady and cloudy during the time when the bud develops, it'll have more anthracene. And this is particularly noticed with the ones where you've got a wash rather than in intensity. So it's also darker when the flower first opens because it fades very quickly. It's not affected by different soils. It's because all of the flower pigments are basically constructed of carbon, hydrogen, and oxygen. There are no other pigments, the chemicals that are involved in that. And carbon, hydrogen, and oxygen, you get all these from the air and from water. They don't come from the soil. Mind you, if the plant is defective and ill because of some lack of mineral in the soil, they're not able to produce a flower, then yes, it'll affect the color of the flower. But you'll also see deformities and you'll notice that the plant is sick. And the sick plant is probably not going to produce much in the way of flowers anyway. Down here, I'm showing you sort of like different concentrations of the filament. So the different concentrations will affect the color and the pigment. Now here's a picture of the plant cell that I promised you earlier. This part here is the vacuole. And it's taking up a large part of the cell place. And actually in the flower itself, the cell vacuole is actually even larger than this. It takes up almost the whole cell. And the definitum is stored all inside this vacuole, which is stored in a water base. So it's in a water soluble form inside this vacuole. The plastids are all in the cytoplasm around here. And actually, like I say, the cytoplasm is lower. A lot of these other structures that they show you in here are more are not as plentiful in the flower's petal as it is in the cells of the flower as they are in some of the other petals, other cells of the plant. So what do the genes do? And I talk a little bit about because people don't understand genes all that well. Many genes are single acting genes in that the trait is controlled by one gene. They're often tracked as Mendelian genes and designated dominant and recessive. And the gene designed as a dominant gene is designated with a capital letter. For example, for PL, small letter is for placata, and you have 4 copies of the placata gene in a placata iris. But if you have one copy of the dominant gene, PL, that results in it not being a placata, and you end up being a purple or a blue, or it has a red, depending upon what the other pigments are going on with it. So placata would have the genetic code 4 copies of the PL recessive, and just one copy of the PL changes it. There are a number of factors of iris, such as branching, bloom time, height, etc., which are multi-factor traits, so it has a number of genes that are involved in that. In modern genetics, we look at genes in terms of what happens in terms of the biochemical or molecular genetics. And you consider the genes as being switches. They turn on or produce a biochemical or process or turn off. So it's not sort of a blending or a mixing of things. It's either there or it's not there. The chemical is either there because the gene's there, or the chemical is not there. So the switch is either turned on or it's turned off. Each of the pigments in iris systems are constructed by a chain of genes. Each gene makes one change and then it's passed on to another gene, which makes another change. And the next gene, if active, it produces another change. Or if it's not active, then the plant stays at that particular at that pigment. Pigment is expressing the chemical is not changed as it remains there. That is, the next gene in the chain is not active, so it doesn't change it to another one. So actually, what you have, and I'll show you, explain that more in a few minutes. The part of the gene, the part of the petal where the gene expresses solid color is controlled under a completely different set of genes controlling the structure or domains in terms of where these colors are produced. The final colors are a combination of the anthocyanin and the carotenoid genes, as well, to give you the mix of colors. And the distribution genes is to show you what part of the flower they're actually in. The interesting thing about genes for the pigment production is that they're conserved across many species of inherited as a common ancestor. So daffodils, irises, etc., and fruit as well, tomatoes, cantaloupe, oranges, all have the same pigment production, same genes. They just have different ways that they're used and expressed. Same genes as beans in the flowers are the same things that we get in fruit. Recessive genes are something which is basically you can consider this recessive gene as a non-active gene, or that's the sort of traditional way, the Mendelian way of looking at it. And that can be true if it's a single active gene, an independent gene. But in irises, we've got a sort of that chain I showed you, and I'm going to show it to you again in a couple of minutes, is sort of like the takes one gene sort of passed on. So a pig iris is designated as a 4 T's as 4 copies of the recessive gene T, as not as a T being a short form for tangerine. And that's not entirely correct, as I'll show in a minute. So the lycopene is a pink that's in the pink iris. We have one more copy of the Y gene, we have a yellow. So the lycopene is now converted into basically beta carotene, or it could be alpha carotene. So you get that change from the pink to the yellow. So one active gene. So we're actually, recessive genes are basically placeholder genes in the sense they're not considered active, but that's not entirely true either. When we call a gene recessive, we need to designate what it is recessive to. For pink, basically it's recessive to yellow, and the yellow designated by the gene Y. So if we have one Y gene, so like 3 tangerine genes and one Y gene, it's now converted from pink to the yellow. the top would be considered, traditionally is considered as TTT, with four copies of the tangerine gene. When tangerine was assigned as a tangerine, it was conceived that the tangerine gene and the four copies of the T converted the carotenoid pigment away from the production of yellow carotenoid pigment into gene, but that concept was not correct. It didn't is one thing at the time. We didn't understand the molecular what's involved in terms of molecular actions of the genes, the biochemical actions, and we didn't recognize that orange irises, and we didn't recognize a lot of what was going on in the genes. So, and there's a number of dark yellow irises which are considered to be tangerine or considered to be orange, and that wasn't correct either. So this is what tangerine or orange looks like. This is the orange iris. This is from my own glaciata seed dune, and I was quite thrilled with this one to get that orange pigment. Now, there's been a lot of confusion about what is orange, and it was confusing. Is it part of it pink or sort of a mix of colors? And a number of people think that the orange is actually mixed with. Actually, it's its own pigment. I'll show you why in the next slide. This is different from what you see with your with the dark orange iris, and there's a number of other factors which are going on there which change the appearance of the pigment. And I'm not going to go into that too much because it can get fairly complicated. This one is a glaciata, which means that there's none of the anthracene in there to affect the color and affect the pigments. The orange is actually a precursor to lycopene and recessive to lycopene pink. There are a number of people who disagree with that, but I'll certainly be prepared to sit down with them and discuss it further if they wish. Now, it turns out that when you look looking at to understand what's going on here a little bit more, I found a lot of interesting information in terms of tomatoes. And basically, I was talking about earlier about the conservation of genes, which basically means the same genes that are operating in tomatoes are the same genes and the same pigments as that are in irises. And there's been a lot of researchers have looked at orange tomatoes, and what they found that in orange tomato, the pigment is actually the tetracis lycopene that's showing you here as a molecule. And the cis actually means that it describes how it bends. So when it bends up, it's cis. When it bends the opposite direction, it makes a straight line and it's trans. So lycopene, which is all trans, has a straight line. So it's the same basically chemical formula, but that bend in here and the bend in here makes all the difference in the world. So you get the orange tomato, or you get the red tomato, depending upon which form is in here. And you can get the orange iris, or you can get the red or the pink iris, because with that thing. And so what happens is you have this tangerine gene is referred to sometimes, or referred to in the iris designation, or the actual name of the gene as designated in the molecular gene, converts that tangerine into the from the cis form to the trans form. So while you call the lycopene recessive, it's not actually recessive, because what it does is it takes it's active changing this chemical into the pink chemical over here. So it's actually being very active. One of the reasons why tomatoes are more intense in color than irises is that it's a fruit and the pigment is entirely through the whole fruit, instead of being just in the iris as a flower. It's just an upper layer and the lower layer of the petal. And so you have the center section, which is probably about half of the flower petals actually doesn't have any of the pigments in it. But there's a second factor too, is that the pigment is stored in the plastids in tomatoes in a crystalline form. So it's more heavily packed. So each of the plastids has more heavily packed. In addition, there's more plastids in the fruit of tomatoes than there is in iris. So there's a number of reasons why that goes on. Here's showing this sort of in demonstrations. So in traditional Mendelian genetics, we have the white and we have the yellow, and they traditionally called the white as recessive to the yellow. So the 4 Ys there, with 1 Y gene over there, they saw the pink as being sort of a side branch off the genetic. And of course, there's no recognition of orange as being a separate pigment. But in terms of molecular genetics, we have to look at this a little bit differently. I'm using CR here for carotenoids, which means that the white here has none of the pigments producing any of the carotenoid pigments. But it also has to be recessive. So it's recessive to any of the carotenoid pigment, but it also has to be recessive for the pigment producing orange. So it has to be 4 copies of recessive. So if we have 1 copy of the recessive or of the dominant orange, it converts the pigments there into the orange pigment, but it also has to have 4 copies of the recessive, has to be recessive for tangerine. Because if we have 1 copy of tangerine, we now convert that orange pigment into a pink pigment. But that also has to be recessive for the yellow, because we have to have 4 recessive yellows. Because we even have 1 recessive, or 1 of the dominant yellow, it converts that into the next pigment of yellow. So we have the conversions from white to orange to pink to yellow. So yeah, we really have to look at these in terms of molecular genetics as having 4 copies. So the pink is not really 4 copies of the T, because we have to have an active gene there, which actually converts that pigment to the next pigment. Now, to make it confusing, we actually have 4 different yellows. But I'm going to show you here a little bit about the designation. So I'm looking here at the designation of the small letter for the recessive and the capital letter for the dominant came right from Mendel, from his original experiments, where the yellow peas were designated with the capital Y for the yellow, and the green peas were designated with a small y, because that's the absence of the yellow. So that's a recognition. The dominant gene is actually converts the green pigment and produces a carotenoid pigment in the seeds, which produce the yellow seeds. So we need to see the genotype of what a pigment is for a gene. This would be, like I say, this is for a tangerine, it would have to be 3 X's. So the X stands for being either of the CR for the producing no carotenoid pigment, or the OG produces a pink. So because if either one of those could be in that position, you have to have the 1T. We use the designation orange and tangerine as a short form for what's called the phenotype, but actually the appearance or the genotype, rather than using we could use the actual gene name, but it's kind of hard to remember those particular genes. So to remember that, we're going back to this chart, and if we're trying to so it's easier to say that we have the tangerine or having the orange gene here, which converts the tetracyclycopene into the all trans pink, so going from orange to pink. So that's actually the Crisco gene, or I'll call it the orange gene, OG. And the LCYB here converts to tangerine into, here we're looking at as a beta carotene. I'm not sure we have this one expressed in the iris, but basically we have down here 4 or 5 or maybe even more different yellows in irises. So you don't have one. People kind of think of the irises as being yellow, and we actually have quite a few different yellow pigments present in iris, and that's something to keep in mind too. But that's not something I go into a lot of detail right here. But again, here, in the chart again, showing you sort of the sequence, the orange gene converting moving it from the white, providing the carotenoid gene, then we're converting it to pink, converting it to yellow. And here's showing you some of the different yellow genes the yellow pigments that are going on. And these are all different pigments. And sometimes you can get one which is called like a near white, And you find this in dailies. They don't have this pure white, they have what they call a near white. So it can sort of vary from one to another. One thing that we find is that having sort of like a broken color with a carotenoid pigment is something which is not really present. You don't have a lot of flowers with that. This is an SDB seedlings by Carlos Atoimus. And this is just something that showed up in his garden this year, and when he's not sure what to make of it, neither is anyone else. And we don't know if it's going to breed true or not, but I encourage him to do some breeding with it. Hopefully it will. Broken Dreams, one introduced by Keith Keppel a number of years ago. And a number of people have used this in breeding, but have not gotten anywhere with it. And here's two more examples where we actually have carotenoid pigments shown in a broken color. Corsage and Joseph Coat. And again, we have not been able to capture this and use this in any breeding in any consistent fashion. But there's an exception here on the screen that's going on, and I've been looking at this one, Cajun Rhythm. And I've been very intrigued with this one because it shows quite a variation from iris to iris in terms of the distribution of the orange pigment in here. So it varies from being almost solid to being part and not quite splashed or sort of splashed, but not quite. So I've used that heavily in my breeding, and I'm going to be talking to that in part 2 because I think there's some potential there for it. Both, but here I'm showing you a little bit of what happens on it, and this is so we can explain a little bit more about chimera and what I'm talking about, the wedge shape. Now, this shows you a petal, and there's like a single point on there. Now, what happens in here is that we start from one petal and it divides into two, and this goes sideways, but also develops lengthwise and sideways. So as these cells are dividing, they're going this way and this way, this way and this way, this way and this way. So we're developing a wedge as we go out. So I'm showing you some chimeras here. And this is where you've got that point mutation occurring there. And then you're having the wedge. So as that single cell goes out along the petal here. And in this case, what we have actually lost is the gene that produces this dark petal. And I'm going to be talking about that at a later, in some of the later slides. So it gets that wedge dividing sideways and lengthwise, sideways and lengthwise, sideways and lengthwise, and going out. Same thing in this one. In this case, we're losing the purple and we're having seen the white underneath it. This one has a one of the yellow carotenoids underneath it, so it has a yellow wedge. Here's sort of like a placata one, and it's showing there's a mix there, but you see some of the placata markings along there, which is not affected by this point mutation, showing that the point mutation is in a gene, and this is again, I said I'll be speaking that later on. And here we've lost the purple gene, and we can see that that dark red underneath it on the spot is actually produced by a yellow pigment. So you see the difference between the purple and the thing is caused by the purple overlay. Again, that wedge. This is one where you have the dominant reduction of anthocyanin, and when that one's removed, you get that purple wedge. So we start with a spot, again, sideways, lengthwise. And this one, we've lost, again, we lost that dominant reduction of anthocyanin, so we get that purple coming in. And where you've got that purple over the yellow, we now see the red. So that gives us some very interesting ideas about what's going on underneath those irises. Two more examples, which are kind of reverse of each other. One which is a white wedge, and one was the purple edge. This one we've lost the purple gene, and this one where we've lost the reduction of purple gene. And two more examples of these chimeras. And two more very interesting examples of chimera. This one is on a placata, and it shows you that the red stitching that you see on the outside of the placata is actually the purple over top of a red. And I think that's very interesting in terms of genetics. Two more very interesting examples of this. I quite like chimeras, and I take pictures of them whenever I see them in the garden. And I usually get a couple each year. And two more examples. Sometimes it can be basically farther down. You can see the same petal on a flower stalk showing up and down. This one has again farther down in there, it shows you almost half the one petal and almost half of the other one. So when it gets half and half. So chimeras are basically development errors. It's a mistake. The plant, when it's dividing, you make a mistake. Now plants are usually pretty good at correcting the mistakes and have all sorts of genetics for that. But it goes by and you get errors in gene replication of 1 in 600,000 to 1 in a million, or maybe 1 in 10 million. So it depends upon how well there's errors and how well they're corrected. Basically, they're one-off point mutations. So it starts from a point and then it spreads out, sideways and lengthwise. They're not repeatable in the sense that you get them and they're kind of an accident. So you get them and you expect that maybe that same plant's going to produce again next year, and it's not. It may not, but it's unlikely. It's just random. They're usually not passed on through increases, although there have been a few examples where you have gotten spores from these chimeras, but it's not very often, but it has to be down in the reproductive cells. They're not part of the plant's genetics, so they cannot be passed on by breeding. They're just on one gene by generally a removal of that gene function. Like I said, on rare occasions, it can be in the somatic tissue and the gene can be reduced on as a spore, but it's not usually the situation. I'm showing you here two examples of references to use, the world of iris and the group iris hybridizers. I'm on here and I'll lead a lot of discussions on genetics. So you're more interested in hybridizing genetics, certainly I encourage you to join that group and join in the discussions. You have to be a member of the group to see the discussions because it's a private group. But if you're not a member of the group, feel free to apply to join